Hello and welcome everybody again to the Scientific Origins channel, where we ask questions about things like evolution and the Big Bang. And are these things a conspiracy by the scientific establishment to trick the average person into rejecting God? Spoiler, no. So we're talking today, we're going to continue refuting all evolution denier tropes, claims, tactics, talking points. And this is a very common one. This is a very popular one we're going to be refuting today. Dinosaur soft tissue. And Mike was so excited about doing this, he wanted to join me for this one. So go ahead and introduce yourself, Mike, and we'll get started.
I'm Mike from Answers in Atheism, and I'm old and irrelevant.
Oh no, Mike, you're not old and irrelevant. That's why the thumbnail for this stream is I was there 3,000 years ago. You're relevant because you were there. You were
right.
a witness.
Yeah, there you go.
You saw it happen.
I was there when that tissue was preserved.
Yes, just 6,000 years ago. No. So we're going to talk about the trope, and we're going to give you the reality first. So Mary Schweitzer is the individual that this trope is based around. You're going to hear her name a lot, and her name is dropped a lot by young Earth creationists, and they conveniently leave out the fact that she disagrees with everything that they say about her work and actively campaigns about it. But they'll name drop her. And it's interesting because she was initially a young Earth creationist. She originally accepted the claim that the Earth was only a few thousand years old. I don't know if you knew that, Mike. Probably did. But she was a young Earth creationist at first. But she was passionate about science and paleontology, so she studied vertebrate paleontology under Jack Horner, who's a very well-known paleontologist. He's pretty celebrated. And you might not know this, but when you work firsthand with geology and taphonomy and stratigraphy and fossil formation, it kind of forces you to accept an ancient Earth. Like this has been mirrored by what's his name? Dr. Ken Wolgamuth. He's a geologist. He's an evangelical, and he also was originally a young Earth creationist, and then when he started studying science and became a geologist, it became inescapable to him that the Earth is ancient. This is a very common story. There's Ken, there's Mary, there's Glenn Morton, the guy who came up with Morton's Demon. A lot of young Earth creationists get into geology and they stop being young Earth creationists because geology is so incompatible with young Earth. So that's what happened to Mary Schweitzer. Sorry, Mike. What did you want to?
Could you please put the name of that Dr. Ken in chat? The last name?
Yeah.
Public chat, so everybody can see it. So that, because one thing on YouTube, when people pronounce things and you can't quite figure out how to spell it.
Yeah.
It's always a problem for people, and people want to do their own research. So we like to give some sources. Loki, what do you have to do with soft tissue? My cat.
Right, yeah. So I put his last name in the chat there. That's Dr. Ken Wolgamuth. He's on Facebook, and he's active, and he responds to people about this issue. He's he's a kind of like an older creationist, but he accepts evolution, of course. Anyways, he's not the subject of the video today. Mary Schweitzer is, and she was with Horner's team on a fossil dig in the Hell Creek Formation in Montana, and they found a huge T-Rex fossil. I think it's the second largest T-Rex fossil ever found, and they nicknamed him B-Rex. And while they were excavating B-Rex, the T-Rex they found, They noticed that the femur was very heavy and also unusually remote. And it was so heavy that it had to be broken into smaller pieces for transport. And once it was broken, Mary noticed an organic scent coming from inside, distinct scent like flesh decaying. And she looked inside and noticed that the inner region of this femur was unusually well preserved. So
Wow.
they transported this femur to their lab, and the material inside, they started to test it and devise a plan. What they did was they extracted some of this material and they subjected it to weeks of complicated and painstaking acid baths using a variety of weak acids that dissolved the mineral matrix in these fossils that was produced by hemoglobin decay, and it rehydrated them. So once they had treated the material to these acid baths, the fragments were possible. It was possible to extract these fragments, and they experimented on them and investigated them. And they discovered that these fragments were mostly constructed of collagen type 1, and they contained a lot of these fossilized cellular fossils, actually. These little rocky cell-sized fossils, osteocyte remnants, and we'll get more into that a little bit later. So that's what happened, right? But what followed was just a huge barrage of misrepresentation. The young Earth creationists are still misrepresenting Dr. Schweitzer, and the misrepresentations go on unrepentantly. They have been corrected many times. Schweitzer herself is vocal about how she detests the way they represent her work and misrepresent her. They conveniently ignore that and just keep running their nonsense. So we're going to get into that a little bit today.
Okay, yeah, this is new information for me on the Mary Schweitzer story. I didn't know about the field story. I'd never heard that before. And I didn't know she used to be a young Earth. So was that when she was young?
Yeah.
Okay.
Yeah.
That happens to a lot of kids, unfortunately. It's a common childhood disease.
We've got Golden Arm here. Golden Arm is a young Earth creationist evolution denier. I don't know if you've met him, Mike, but he hangs out. He hangs out around some of the open panels and talks on them sometimes. So hopefully you're going to learn some stuff today, Golden Arm.
Maybe he'll talk to me someday. That would be really fun. I love talking to evolution deniers.
He might. So that's it for this slide. Let's go to the next one. So that's the reality. Here's what creationists say about this subject. These are some of the tropes that young Earth creationists say about Schweitzer's discovery. They'll claim that we found wet, unfossilized muscle, fresh liquid blood, and red blood cells sitting inside this ancient T-Rex femur. And because these things can't possibly stay preserved that long, that means that T-Rex extinction was much sooner than the evolutionists say, and actually the young Earth timeline fits. Well, the first problem is what they found were fossils. It is untrue to say that fresh liquid blood, unfossilized muscle, and red blood cells are in any way what they found. That's not what was found. What was found was hardened rock, remnants of living tissue that had been converted into rock and mineral over time by typical taphonomic forces. So a fossil was discovered, not a bone with blood and blood cells in it. So what happened is Schweitzer, like we said, demineralized this mineral matrix, which was produced by the hemoglobin decay, using these acid baths. And it took weeks of these acid baths, and what was left behind was elastic microstructures, flexible extracellular protein scaffolding, which is made mostly of collagen type 1, and cellular ghost of osteocytes, not rotting fresh meat.
Right.
So what is an osteocyte? An osteocyte is it going to pop up?
Bone cell.
Yeah, there we go. Osteocyte begins its life as a cell called an osteoblast. And an osteoblast is basically a bone-building cell. It secretes a soft protein matrix that helps bones grow and develop. As a result of this, though, sometimes these osteoblasts get trapped in tiny microscopic chambers in the bone called lacunae. So this helps you begin to understand how these things could be preserved so much longer than usual in these unusual circumstances, because they're already ensconced in one preservative matrix, and that is the bone matrix of the lacunae. But then they get ensconced in a second protective matrix that's produced by the iron ions given off by hemoglobin as it decays. So when osteoblasts get trapped in these lacunae, they mature into osteocytes, these bone cells. And Schweitzer found tiny little star-shaped microstructures. She was very cautious about calling them osteocytes. At first, she was just referring to them as these structures. And her team, do you know how her team confirmed that they were actually osteocytes, Mike? You ever read about that?
No.
So they used protein identification. They used mass spectrometry and immunology to detect specific structural proteins that make up vertebrate osteocytes, namely actin, tubulin, histone H4, and PHEX, or FEX, or PHEX. And that last one, PHEX, PHEX, is a protein exclusively produced by bone cells in living animals. So it ruled out bacterial contamination, antibodies, and artifacts. It ruled out everything else that these things might be. And so that's part of how they were able to confirm they were osteocytes. I just thought that was really cool.
Okay.
So she found these, and it's important to note that they were not cells anymore. They were basically fossilized. They weren't living cells with fluid inside of them and functional organelles or even intact membranes. They were cellular fossils. You might not know this, but it's possible for even cellular organisms to mineralize and become a fossil, and that's exactly what happened with these osteocytes. So this is how the things actually survived. So evolution deniers are going to say these fossils can't be this old. How can this stuff survive this long? So the Earth must be young. The reality is that these fossils survived because, like we've been saying, hemoglobin decays over time, and as hemoglobin decays, it gives off iron ions, these Fe2 plus ions, these single molecules that have slightly positive charge because they've lost 2 electrons. And those lost electrons and their molecular geometry makes them really strong catalysts for hydroxyl reactions. So these hydroxyls are produced by the iron runoff, and they form a linkage. They form a tessellating polymer, and the polymer basically works like a chainmail coating around the already bone matrix ensconced osteocyte remnants. So they're protected by the bone matrix, and then they're also protected by this this hydroxyl and iron ion polymer and it basically works not unlike the way like tanning or formaldehyde works. It's basically a natural preservative. And Schweitzer's team tested this. They put blood vessels in water and they dissolved in days, and they put blood vessels in concentrated iron and they lasted for more than 2 years. And we're talking about blood vessels, not osteocytes ensconced in a bone matrix. So it's really, really strong evidence that iron protectant can be a very highly stable and decay resistant shield to biomolecule remnants. So that's how those things actually lasted that long.
Okay.
Another trope, they claim we found dinosaur DNA. No, we didn't find DNA. If you want to call them on this, ask them for some AT, C's, and G's. Ask them for the sequence of this dinosaur DNA we found. I guarantee they can't produce one because we didn't find any. There is no ancient dinosaur DNA we've ever been able to recover. What we have been able to recover are biomolecule remnants. And because molecular biology is a thing, we're capable of doing molecular biological comparisons even without a full DNA strand, a full long strand of DNA that can be sequenced. We can compare the biomolecule remnants we have to modern biomolecules, and we've done that. And interestingly, which do you think has more similar biomolecules, Mike? Do you think a crocodile has more similar biomolecules, or do you think a chicken does?
What? What?
If you take the ancient biomolecules you can get from some of these dinosaur fossils and you compare them to modern biomolecules, which species do you think has more similar biomolecules, a crocodile or a chicken?
This I don't know. I'm guessing they're the same.
It's a chicken, actually. It's a
Really?
chicken. It's all the birds. Birds are
Why?
closer than they are to reptiles because birds diverged from the common ancestor they share with dinosaurs more recently.
Oh, you mean the DNA sequence?
No, they did biomolecule comparisons. We don't have DNA. We have
Okay.
biomolecule remnants, but you can still do biomolecule comparisons instead of DNA comparisons. Similar to comparative genomics, it's just comparative molecular biology.
Yeah, let's put a bookmark in that one because there's something that I am not getting about that because biomolecules are the same across all of life.
There's a paper I can send you on it.
Okay. Yep.
Yeah, so alleged early reports that there was ancient DNA found in things like amber, those were disproved. They were shown to be modern bacterial or human contamination.
Yeah, there's something really important here.
Oh,
Now, if these things were less than 6,000 years old, we would find DNA in a lot of them. It'd be all over, and we'd be able to sequence that DNA because we have sequenced DNA. I think as old as like 700,000 years or even 1,000,000 years old. We
that's
found
correct.
sequenceable DNA in Neanderthals, for instance, which are over 40,000 years old. We sequenced the DNA. In none of these dinosaurs that they claim have soft tissue and DNA have we ever come up with a sequence. What we come up with is remnants, basically. And now here's the thing about remnants. It's really important for the creationists to understand. When a Animal dies and gets buried. It has biomolecules in it, right?
Mhm.
Those biomolecules don't evaporate because they're stuck in the mud. They're basically buried in a non-oxygen environment, and they're stuck there, right? They turn into something else, as things do over time. When one organic chemical turns into something else, guess what? Uh, they don't do it at the same rate. Every organic molecule has a different rate of basically composition in these closed off, non-oxygenated environments. Collagen and heme happen to have the longest. And porphyrins, chlorophyll, remnants from that, you'll find that in coal. So the important part is you have to ask yourself, what happens to the carbon? Does it evaporate? No, it does not. It's there somewhere. And there are pieces in different small degraded carbon compounds, depending on what the starting material is. So ask yourself, what is going to happen to the animal when it decays? It doesn't evaporate. God doesn't remove it. It's still there.
Yeah, minerals flood into empty spots, and that's
Right.
pretty much what fossilization is.
Now, we've known in coal for years that there are porphyrins in coal and remnants of chlorophyll in coal after 300 million years. And we've known this since 1930s, I think, or
Yeah,
40s. Basically, we've
DNA
known
in
this.
typical conditions only lasts about like 521 years, but
Yeah.
DNA when it's frozen can last up to 7 million years. So
Yeah.
we've gotten DNA from frozen samples.
Yeah. And coal is developed at a highly acidic environment with very low oxygen because of peat. And there are all kinds of preserved biomolecules, remnants of biomolecules, particularly these complex ring structures like hemoglobin and chlorophyll. And all of that stuff we have known for years. It's never been a surprise. And we sure as hell don't call it soft tissue. We call it
Yeah.
coal.
Yeah, so this is my last slide, and I think you said you have some stuff you want to bring up and screen share too. This is just summarizing some of the most important points, some of the things you're going to hear the most. Uh,
Yep.
Evolution deniers, young Earth creationists are going to claim fresh blood and meat were found. That is a myth. Osteocytes are microscopic structural cells that were embedded inside of a mineralized matrix. That they didn't find soft, fleshy muscle tissue or liquid blood. Another myth, we found dinosaur DNA. Again, this is a myth. There's no there's no ancient non-avian dinosaur DNA sequences available anywhere because there wasn't any DNA ever found. All those samples are way too old to still have DNA in them. Interestingly, a point of nuance here, we don't date fossils based on how well preserved they are, precisely because there's a bunch of factors that play into how well preserved a fossil will be after a given duration. We use other methods, and another method was used on this that it wasn't radiometric dating. It was a similar chronology technique, but not exactly that. And that dated the femur from B. rex is very, very old, of course. And yeah, that the last point, DNA can't last that long. So non-avian dinosaur extinction happened much more recently than evolutionists claim. You're right that DNA can't last that long, and it didn't. So that's why we didn't find any. So that covers my slides. If you want to screen share, I'll go ahead and remove this from stage. There are a couple other points, of course. Me and Mike were talking before we went live about how basically what Mary Schweitzer did is she inadvertently created a whole subfield of science that you could call like unusual fossil preservation studies. And so it's a lot more complicated than what I presented. And Mike's got some other stuff he's going to share too. How do I make you full screen?
Let's see here. I've got
there we go.
Got that paper there, but we're not going to look at that yet. We're going to. I should have been ready for this, Austin. I'm sorry. Let's see here. Where are we at? Thomas. Okay, so first of all, I want to mention this, and this just came out. It's a Science Daily article, and they pick the clickbait where they said basically, oh, come on. 1 in a million sea creature fossil preserves soft tissue for 450 million years. Now, here's the other thing. When you hear that soft tissue was preserved, you have to ask yourself, what do they mean exactly by preserved? Okay.
Oh,
and
real
in this
quick,
article,
Mike, one quick thing. It was amino racemization. That was the dating technique they used.
okay.
So, yeah. Sorry, go ahead.
Yeah, and that one has all kinds of problems. I've looked into that extensively about 6 years ago, and I have a whole folder on that. It's not really a very good way to date things, actually, they've determined, because it's all over the place. Anyway, so here's the headline, right? And this sounds like, soft tissue, 450 million years old. How could that be? It couldn't possibly be that old. It's got to be less than 6,000, right? So you go through this whole thing, and they mentioned the paper. Oh my goodness, why are you doing this to me? Fossil record, okay. And the paper, the actual paper, is no better. They do the same thing. They use the headline article. I'll make this smaller. Okay. Oh my goodness, it's still doing it to me. Okay. Exceptional soft tissue preservation reveals the oldest evidence for 2 feet in their ecological significance in crinoids. Okay. Hang on a second here. Okay. So that's what they chose for the article. But then in the abstract, they tell you. Now, the importance of this article is not that they found soft tissue. That's not why they wrote the paper. That's not the biggest discovery. The biggest discovery is they were able to find crinoid feet from 450 million years ago and determine that they were different than the extant crinoids today, that there's been some evolution there, right? The tube feet are preserved as pyrite films and exhibit morphology that is broadly similar to that of extant comatulid crinoids, but with combinations of length and spacing that differ from those documented in extant crinoids. So that's the big discovery of the paper, that they found this structure. So what they mean by preserved, and this is the same thing with the osteocytes, what they mean by preserved is that there was an impression of this soft tissue that was left in the mineral composition, right?
Mm-hmm.
So the soft tissue did not survive. What survived is if you press your finger into a piece of clay and your finger rots away entirely, all the bone, all the finger, it's still going to leave the impression in the clay mineral, right? And that got preserved. This is preserved in pyrite. Now, those osteocytes and those blood cells, it's the same thing. Those cells are structures. When they die and basically get buried, they stick around long enough to leave an impression that we can find with a microscope. And they'll say, oh, they found osteocytes and they found blood cells. They didn't really find anything of the sort. They found impressions left from the structure of the biological tissue that was there. Right. And as we get better and better with electron microscopy and CT imaging of these fossils, we are finding more and more of these very, very fine details of biological organisms. So that's important.
I think it's a very similar case to the DNA remnants they found. It was like They found debris, nucleotide debris inside the osteocyte remnants. Trying to find.
What is this petite flower? What I've heard her say is that she hasn't been able to disprove that it's remnants of the original material. You can disprove that by doing a basically chemistry. You can figure out exactly what you have, what chemicals are preserved. And Mary Schweitzer, of course, had actual collagen preserved, which is another story. It's a different story. Those aren't impressions. Those are actual collagen biomolecules, and we'll get to that. Okay. But now there's another thing, hydroxyl radicals, and this plays into collagen. Collagen is an interesting little protein. It has these triads of amino acids with glycine, proline, and hydroxyproline, which is basically hydroxylated proline amino acid. It's not the same as your usual amino acid. And collagen forms this trimer. There's 3 molecules that wrap around each other. And oftentimes it'll be 2 different kinds of collagen. She's talking about collagen 1. And that structure is the most stable biological material we know of, collagen. You're made of about, I think it was like really like 30% collagen. That's what we're made of in proteins. And it supports our body, and it is like a super, like copper braid, a braided
It's one
molecule.
of, if not the most robust biomolecules.
Right. And they have been able to detect little pieces of that collagen and actually determine the protein composition, because we know they turn into they're not proline. They might be glycine yet. They're not proline and hydroxyproline. They turn into something. I think hydroxyproline they find, and the other 2 become like they convert to some other organic compound. But anyway, this is and this paper here. This paper goes after young Earth creationists, and I can put this in. I'm going to put this in the chat. They go after the whole soft tissue thing. Hydroxy radicals are both useful and highly toxic to the body. They're involved in the activation of certain proteins, tumor suppressor during chemotherapy. However, if unchecked, hydroxy radicals in excessive amounts can cause significant damage to biomolecules, right? However, despite the destructive potential of hydroxy radicals toward biochemicals, a healthy body has a variety of mechanisms that prevent it. Protecting, let's see, plasma. Oh, why did I want to read that? Maybe I haven't read this article yet. I'm sorry. I'm just doing this on the fly. I didn't have much time to prepare. Such scavengers also include type 1 collagen. It's a type of collagen in CBM and in tunica adventitia, the outer layer of connective tissue of a blood vessel. Its long fiber shape provides that after hydroxy radicals have caused conformational change in the collagen by reacting with its amides, more residues, oh God, this is dense, enabling them to react and scavenge more hydroxy. The presence reduces the generation, and I believe that causes the preservation of collagen. Phospholipid membranes of cells have built-in protection. We got to read this and figure it out. But anyway, so that's something else to consider. Collagen is preserved. They can see the chains basically because the chains are in place, but there's no longer the original amino acids or proteins. They have been degraded for millions and millions of years, but they have left this skeleton, this remnant. There's something that we can look at, and that's what Mary Schweitzer found. She found, actual collagen remnants, and those collagen remnants, they become extremely cross-linked, all the amino acids, and that helps basically preserve the protein structure or protein remnants. So you gotta, like, our language, soft tissue was preserved, can mean a number of things. It can mean an impression was preserved, or it can mean some actual remnant of an organic molecule that was there. And again, you have to keep reminding yourself, organic molecules don't just evaporate into the ether. They turn into other organic molecules, degraded organic molecules. So anyway, what are we finding? Yeah, what did Sydney say? Collagen fragments remains were dry and only exposed when samples soaked 3 to 4 weeks in etching acid and rinsing. Yeah, they found they got rid of all the minerals, and they found these remnants of organic compounds that had broken down, and that was a surprise. Yeah, we were surprised. It's like, whoa, but it's a good surprise because we can actually compare some of those proteins and start to work out a phylogenetic tree based on collagen, which they'd done a little bit of work on. So,
yeah, it's really cool, Golden Arm. So hemoglobin contains iron. As it breaks down, these free radicals get produced, and the iron is a really strong catalyst, and they can form. It's really common for ionic compounds to form lattices because it helps them distribute their charge effectively. And that lattice basically forms a protective layer, keeping these things from degrading as quickly as they normally would. And like I was saying, they tested this using more degradable material, the blood vessels, and they were able to preserve them for over 2 years in an iron concentration, whereas in a water solution, they degraded in just a matter of days or weeks. Yeah, and also, Mike, when you were talking about like the remnant fossils, that's kind of what happened with the DNA remnants. They use staining agents on these osteocyte remnants, and the staining agents showed these faint signals that resemble localized nucleic acid debris, and they were able to use that debris to approximate what the DNA biomolecule would look like, and then they did comparisons with that.
Yeah. This is a porphyrin ring. Basically, it's a complicated organic ring. At each of these little vertices here, there's a carbon atom, and that's a double bond right there between 2 carbon atoms. Then we have a nitrogen in the middle. They're basically, this whole ring is extremely well preserved. And lasts for millions of years. And we found them in coal. And we found all sorts of compounds like this in coal that either the the remnants of chlorophyll and even heme. And this is just something that we've known for years. And here's some other structures that form. And when these carbon structures form, they're kind of like graphenes. Those new nanomaterials are making that extremely hard.
Yeah.
And remarkable. Carbon does that. Carbon's capable of some amazing preservation. And
that's right. We're getting hard for the dinosaurs over here. Golden Arm, yes, this type of preservation is found in other animal fossils. We've never recovered any DNA from dinosaurs or anything as old as them, and we don't expect to find DNA from these soft tissues, but we can always hope. DNA has been recovered, however, from samples more than 10,000 years old, even more than 300,000 years old.
700, I think, was the oldest.
If dinosaur fossils were as young as creationists claim, finding soft tissues and DNA in them, it wouldn't be news. Recovering DNA from them would be easy enough that it would have been done by now. So,
yeah. Yep, and yeah, she did some stuff with iron is another preservative. The thing is, like, I used to be really fascinated with this in organic chemistry, and that is the decomposition of plant material. And that's why I knew that about coal. And it's really interesting. When things decompose, they leave remnants. They leave they leave a causal structural chain so that you can find weird little organic molecules, and you can inductively figure out what biomolecules they came from. They're still, they're not biomolecules anymore. That article I just had does a little bit on lipids, and I wasn't aware of, because that would be, the lipid bilayer on the outside of the cell. That's going to leave the impression of a cell. So microscopically, you can see where the cell structures were, because, all that stuff decomposes at a different rate. It's going to mineralize at a different rate, and you're going to be able to see things that look like cells, but they're not cells. If they were cells, they would get up and eat you, right? The T-Rex, right? Don't you think?
Yeah.
Austin?
Yeah, right.
Yeah. We don't want that. Paleontologists want in the field, and T-Rex just eats them.
Environmental DNA. That's cool.
Huh?
Oh, sorry. I was just muttering about Sydney's comments.
The oldest DNA, 2 million years old. Okay. Recovered environmental DNA from Ice Age sediments. Cap Colbon Formation of northern Greenland. Cool.
And environmental DNA, that's exactly what it sounds like. Probably some living organisms spat or urinated or defecated in that area, and some of its DNA was projected into the environment. This is a very real thing. Isn't it? That is pretty cool.
Is Sydney Raptor some kind of a walking encyclopedia?
Kind of, yeah. Freezing is better at preserving DNA. So DNA decays around 521 years in standard conditions. In frozen conditions, DNA can last up to around 7 million years.
Wow. Okay.
I'm not sure. I think you'd have to ask Sydney that. I wasn't familiar with it until he brought it up. But it's a good question.
We don't know what's from.
The environmental DNA.
Oh, okay. The stuff that they found in the layers up there. We know it's from they've been able to work out ecologies, basically. It's from all the animals and plants that were there, and the DNA just got preserved. And we were able to figure out a hell of a lot of stuff, which is kind of cool. But we do know roughly what phylogenetically what they were related to, and we've been able to work out ecologies with that. Okay, porphyrins really have been isolated from coal. So, okay, porphyrins and coal. There is our bonnet. I'm pretty sure some of these articles are pretty old because I read about it back when I was like in my 20s, I think. Let's see. Coal is not just elemental carbon. It is an enormously complicated organic macromolecular material derived largely from plant matter. During coalification, much of the original biological material decomposes, but chemically resistant portions of plant biopolymers can survive and undergo further cross-linking, aromatization, reduction, oxidation, and rearrangement. Okay. Hatcher and Clifford describe modern coalification models, and there's been a lot of research done on that too, of course. And we have pyrroles and
this is the fascinating intersection between chemistry and biology, right? Because all living things are made up of the same substances everything else in the universe is, and that they have molecular geometry that tells us a lot about how they act, and that helps us find out stuff about ancient living systems. And I just think that's so cool.
Yeah, and the whole thing, taphonomy, the study of fossilization, it's probably a field in its infancy. We're just, now that we have these new techniques and all of, electron microscopy and all of that, we are learning new things, right? Basically, and there's no surprise here. And it doesn't say that the dinosaurs were 6,000 years old, because if they were 6,000 years old, Petite Flower and everybody else, we would find DNA, basically.
The age of the fossils isn't determined by how well they're preserved, like I said, because preservation depends on far more factors than just age. And the age of this particular bone was determined from the age of the rocks it was found in, the Hell Creek Formation, and that formation has been reliably dated by several independent methods. So even if you have an issue with the racemization dating. They also dated the stratigraphy and used other chronological methods. So really reliable range of age for that thing.
Yeah. I've got a whole bunch of work. Like I said, what did I have? Like 20 different articles that I've gone through. And this is a field in itself that would take me months of study. But of course, I'm working on embryology, Austin, at your
Get
behalf.
to it, Mike. You gotta have one screen open with embryology stuff and the other screen open with taphonomy. You gotta do this stuff for me.
I'm almost through the third week of human embryology, human development. Almost 3 weeks, right?
Yes.
It's a weird looking little disc at that point.
Yes, biology is the science of icky, squishy things. Yeah.
Yep, there's a whole lot here. We come up, we're not at an hour yet, so Robin starts at 3 over on skeptic philosophy.
Mm-hmm.
But yeah, my God, I don't even know where to start here.
Yeah, the reports of soft tissue, like, although they're really remarkable, they've been sensationalized even further. That's basically what's happening. The evolution deniers are like really over sensationalizing this. The tissues weren't soft or pliable originally. They had to be rehydrated and removed from the mineral compounds. And once they were, you could move them around and they'd be flexible tissues. The nucleated cells have been found before in which the original material was not preserved. So it's yeah. Oh, thanks, Big Bad Mama. I appreciate your support very much. You have one of the best evolution denial refutation channels on YouTube as well, my friend.
And the reason for all of this, of course, is this ridiculous shit where they have a headline like this, and people see the headline, and they don't go and read the paper and actually figure out and ask questions. What was it really? What did they really find? Did they find soft tissue that was 450 million years old?
Right.
No. It was iron pyrite, which is fool's gold. That's what they found, pyrite films.
Yeah.
They didn't find any organic molecules that they're talking about. So over and over again, we keep running into this. The popular science press shoots us in the foot every week.
Yeah, it's tough. They need to be more responsible about the headlines they write, but I think generally they just aren't aware that there's people out there that will weaponize the things they say. And yeah.
Here's all the reasons why collagen can last. Iron cross-linking, encapsulation, and mineral matrix. There's this whole thing about encapsulating in these little clay pockets. I think it's called CBM or something like that, clay something or another. That's another preservation method for biomolecules. Dry and anoxic environments, Maillard reactions. Why it should not last, protein decay rates, environmental exposure, contamination, misidentification. What's actually been found, mass spectrometry has identified collagen-like peptide fragments. Peptide fragments. It didn't say fully formed proteins or fully, or one of the 20 amino acids.
Yep.
They're
no DNA.
Yeah, they're typically very short and degraded. Immunohistochemistry, you were talking about that. I think that's how they might find the DNA.
Yeah.
Shows binding to antibodies for collagen, but then that's false positives. What else we got? Collagen lasting millions of years would be extraordinary, and extraordinary climbs require extraordinary evidence, of course. What else we got? Okay, here's a table. Let's see. Here's a detailed table. Collagen decay rates, which goes through a whole bunch of shit, and it's complicated. But if you know anything about chemistry, you realize that there has to be remnants of those biomolecules. They're all there. The whole damn dinosaur is in that little pocket of minerals somewhere in some form. Some of it may have evaporated as carbon dioxide, but not for the most part.
golden arm, it says that it's abnormal, and then it gives a bunch of mechanisms for how it can happen, despite it not being a
Yeah.
typical occurrence.
Yeah, it
But
isn't going to happen. You're not going to find it. And unfortunately, I think they're not looking enough, because the more we look, the more of this stuff we're going to find, the more clues. Now that we have these advanced techniques where we can get down and look at tiny little things, we could learn a lot by checking out a lot of dinosaurs. But you don't really want to be cutting up every dinosaur fossil, right? Because you're kind of like ruining them when you do that. So
yeah,
yeah,
and so if we want to start wrapping up, I think I have a good final point we can end on. If there's anything
sure.
else you want to go on, get in before we do, let's do that.
Make me small again. Yeah, and the other thing is, you don't just find something new in science and say, "Oh my God, young
Right.
Earth."
Yeah.
You don't just conclude. You look and you look and you look, and what we should do is we should actually drive out there to where they found it and start asking them questions and have them show us on a spectrometer exactly what biomolecules they found, because they sure as hell aren't going to be collagen proteins.
Right.
So I mean, you can't just find one little one-off thing, golden arm, and say, "Oh my God, that solves the problem." No, it's a lot deeper than that. Look,
Yeah, you
think,
have to
work.
test out other explanations. You have to uh, collect more data. You have to make sure that you're not just encountering an outlier. And we pretty much verified that this is an outlier. It's possible, but it is still an outlier. It's something that isn't common, but it can still occur through explicable mechanisms.
Yes, that's ChatGPT. That is a website where I do most of my research, and I just ask it questions, and drill deeper and deeper and deeper. If you do the same with an open mind, you will find that this is another big scientific story, and I encourage you to go out and do your own research.
Yeah, so the point I think we can conclude on is that if the soft tissues were the validating evidence that young Earth creationists and evolution deniers claim they are, you would be able to recover DNA from them. So
Yes,
until we get dinosaur DNA, this talking point is dead.
yes.
So thanks for stopping
Why
by,
hasn't yeah.
everybody. I hope you enjoyed it. Golden Arm, it was fun having you come by and do a little pushback. I hope you learned some stuff. And if you're watching this, be sure to like, comment, subscribe, do all the social things, share it with your friends, share it with your significant other. Go to your local tavern and demand that the barkeep put it up on the TV for everybody to watch. Just do whatever you got to get this information out there, to get these conversations going between people who know what they're talking about, so that it's productive. And yeah, thanks everybody for stopping by. Thanks Mike for coming by. Thanks everybody in the chat for being great members of the chat. Sydney Raptor with all the facts and the relevant information. We love the support. Anything else you want to say before we close it out, Mike? Or is it goodbye, cruel world time, do you think?
Just a little comment about AI lying to you. No, AI will get things wrong, and it will sometimes just make shit up, pull it out of its butt. But if you keep asking it questions, if you look at my AI pages that I was scrolling by, they're typically 50 or 60 pages long, because I drill down on every single point. I have it give me references. I go out and check the references. I ask it more questions. It's called curating AI.
Yeah.
If you know how to use AI,
Always double check things. I've primed my AI to only give me things it can back up with highly reliable resources from the peer-reviewed corpus of primary scientific literature, and that's a good thing to do. The problem is AI can hallucinate sources, so you always want to double check things. Yeah.
Yeah, and if you know how to use AI, it's like any tool. If you using a crescent wrench, if you're working on an automatic transmission, it can slip and it can give you a serious wound.
Yeah.
AI is the same thing. It's a tool. You have to learn how to use the damn tool, and always think and always dig. There you go. Okay, that's all I got. Goodbye, cruel
All
world.
right. Goodbye, cruel world. Thanks everybody for stopping by.